have to describe what the motion is and how and when it's bad and when we make it better. So I'm going to see if I can get things going here, see if my videos can work. Okay, this is a wild type chlamydomonas. It's actually not quite wild type or normal. Okay, can anyone guess what's wrong with it? Okay, it doesn't have an inner ear infection, but it only has one flagellum. Normally they have two. Okay, and we like it because if it only has one, it swims in a circle. You know, kind of like I would be if you just nailed my foot to the ground. But that helps us study it because it doesn't swim out of the field of view. It stays in the field of view. We can analyze it. Okay, this is a mutant that's lacking a certain motor protein inside its inner arm. It doesn't look that different, except it swims a lot slower. Okay, now people, if you saw these two types, these two phenotypes in cilia, you wouldn't be able to tell the difference because they're both doing the thing. One of them's not dead in the water, it's still motile, and it's not doing, you know, it's not obviously spastic, but it is not as effective. And so it's our job to try and analyze what's wrong. So we do a number of things. We do some standard image processing to keep the flagellum still. Let's see if I can find my mouse. Oh, there it is. Okay. So we make the, instead of rotating around, we make this guy stand still, and we use multiple beats to produce a typical motion. And there's a little bit of jitter because we're taking multiple beats and combining into one slow motion beat. And then we're mathematically describing the shape of the flagellum by a simple mathematical function, which is the same mathematical function that describes the bending of a beam, a normal beam. Okay, this is the normal wild type, and this is the, where's the mutant? Let's see. There's a mutant right here. Okay, so it's the same thing, but it's just different. Right? And again, it's doing the same basic things. There's a bend, it's propagating outward, and it's pushing forward. You can see a reverse stroke, a recovery stroke, and a power stroke, just like you would use if you were doing a breaststroke or something like that. But one of them is not doing it as well as the other. The analogy I like to use is one of them is like Michael Phelps, and the other is like me. Okay, we both do the same strokes. We both do like this, but he's going about 2 or 3 times as fast, and it's important. You know, the subtle things that are different. So we stop the motion, we slow it down, we freeze it at certain points. Here are 3 wild types. They're obviously got a longer stroke. Even though they're doing the same thing, recovery and a power stroke, they have a longer, more effective power stroke. And we can analyze it, and these are what the 3 mutants do. Again, it's very repeatable across different individuals in the 2 groups. Okay, so we can do statistical comparisons. It turns out these 2 mutants, in terms of beats per second, again, they're doing the same thing. They're taking the same number of strokes per minute. Now, I can do the same number of strokes per minute as Michael Phelps, but in terms of how effective they are in going around, you know, Michael's going to go twice as far as I do in the same amount of time, and the wild type goes twice as far as the mutant. So normal and mutant flagella beat at similar rates. Pediatrician wouldn't be able to tell them apart. No offense to the pediatricians, but they would look the same, but they would lead to dramatic differences in propulsive effectiveness. So we do studies that examine the movement of primitive microorganisms, and these studies have already provided insights into the differences and the distinctive functions of the dying arms, which are the difference in the two mutants. So, you know, just the things to remember, the things I would ask you to take away, is that cilia and flagella, they're complex biochemical, mechanical systems, and sometimes mechanical engineers can help understand those types of things. Motion occurs as a repeated propagation of bends, and these mutations in the inner dining arms influence the bend shapes. And these inner dining arms are like the motors, the muscles that drive the coordinated activity. So, you know, how this relates to pediatric disease. Tom's population in the Amish with PCD, that's a rare disease, but there's many common diseases that we think and are likely caused by cilia dysfunction. Perhaps not the severe dysfunction that Tom identified in PCD, where they just don't beat or they beat with straight arms, but perhaps they beat less effectively. Okay? And clearly genetics may play an important role. This group is seeking the disease-causing mutations and doing comprehensive genetic testing, but we're also, you know, seeing how well they work, examining the phenotype. seeing how well they work as swimmers. So we think that using these models, we can not only evaluate the function and gain understanding in the disease, but we'll be able to use these to evaluate the effects of interventions like small molecule drugs on chlamydomonas. So we just want to, there's a lot of people to thank, as well as the team members. We enjoy weekly meetings or bi-weekly meetings. We've been able to hire, with CDI support, a couple of WashU graduates that are very talented. Brian Lewis is a biology graduate, now going to an MD PhD program at Case Western. Shauna Crawford is actually a woman. Shauna Crawford is a biomedical engineering student who is now just graduated from WashU and is working in our lab. And a number of undergrads and other staff have participated. And it's been a great project, and the support of the Children's Discovery Institute has been a terrific benefit. So thank you, and Tom and I would be happy to answer questions for as long as they'll give us.